Okay, welcome back, everyone. I hope you've had a good break. We've got a set of 6 speakers in this session. We've got Carolyn Chong, who's going to go first, Alexander Schmidt-Lebuhn, Lyndall Hardstaff, Rahman Jawad, Abdul Rahman, and Nathaniel Kingsley. Without any further ado, let's get into it. And Carolyn's going to talk to us about ex situ conservation of a critically endangered fern. Thanks, Carolyn.
Thanks, David. Hi, everyone. I'd like to acknowledge and pay my respects to all traditional custodians on whose lands we are meeting virtually and living today. And I'm coming to you from up northern Larrakia Nation country. And today I'd like to give you an overview of an ex situ conservation project I led while part of the National Seed Bank team at the Australian National Seed Bank at the Australian National Botanic Gardens in Canberra. So we're all well aware of the multitude of threats facing biodiversity globally and the role of national and international targets for ex situ plant conservation towards preventing biodiversity decline. And now the utility of our insurance populations of plants and seeds really depends upon collecting and maintaining germplasm that is representative of the diversity of wild populations, and it depends on understanding the ecological and biological requirements for species to germinate, grow, and survive. And so recent reviews of the Global Strategy for Plant Conservation Progress Against Targets to 2020 highlight that particular groups, including ferns and allies, are particularly underrepresented in our conservation banks. And so investing our efforts in increasing our expertise and capabilities in ex situ conservation of these groups is a significant contribution towards our ex situ conservation efforts and targets. Our case study species is a large terrestrial fern, the Dales waterfall fern, Pneumotopterus truncata. It's nationally listed as critically endangered, and within Australia, it lives only on Christmas Island. So the overarching objectives of the project are to develop protocols for ex situ conservation of the species and to establish an ex situ insurance population, and in doing so, support in situ conservation management. And this project is a collaboration of many individuals and groups working together extremely well. So funding was provided by Parks Australia Executive, and the main players are the National Seed Bank, the Australian National Botanic Gardens, Christmas Island National Park, and ATH in Cairns. Bring straight over to the island. So Christmas Island is about 1,500 kilometres west of the mainland. And the island has a unique geological history. So it comprises a limestone cap created by coral reefs over a volcanic basalt seamount. And Christmas Island National Park is situated in the west of the island. Now, the particular habitat of the Dales waterfall fern within Christmas Island National Park is it's situated within a Ramsar listed wetland site, the Dales. So there's multiple threats facing the ongoing survival of this species, and in particular, Parks Australia monitoring has demonstrated extreme fluctuations in total population size, along with low population numbers. So I'll now take you through a bit of an overview of the workflow of this project from collecting to experimental trials. Firstly, following monsoon season, which was March, we did maternal line collections from 2 sites within Christmas Island National Park. So we collected whole fronds bearing mature spore into large paper bags and maintained these at ambient conditions to facilitate spore release. So following spore cleaning and packaging, we transported the material under biosecurity permit conditions from Christmas Island to Australian National Botanic Gardens in Canberra for ex situ trials, and short-term storage material was in a fridge in the dark. So as mentioned, a particular feature of the habitat of Pneumotopterus truncata is it lives on extremely alkaline limestone rock. So we were particularly interested in understanding whether there were interactions between germination media and germination success and early growth. So our germination trial conditions included replication across lines and sites of 4 germination media, including sphagnum moss, moss and sand mix, a water agar, and an alkaline agar. Spore was hand sewn onto sterilized plastic containers and sealed for the germination phase, and incubation was done in a seed incubator. At 2 to 3 months, germinants on agar were transferred to sphagnum for further propagation, which was conducted in greenhouse at Australian National Botanic Gardens on capillary matting. And at this stage, the lids of the containers were opened to facilitate gas exchange and water flow. And it was at 7 to 10 months that sporophytes were transferred to pots to grow on. So overall, we achieved germination success from 15 of 16 maternal lines tested from the 2 sites. And time to first observed germination was approximately 4 weeks on agar and 6 weeks on sphagnum. There was seemingly significant effects of germination media, both on total % germination and time to first observed germination, with both the fastest and greatest germination achieved on plain water agar. There's also a significant effect of maternal line on overall % germination, indicating that different individuals have different reproductive or germination success under the trial conditions. There's also noted but not unexpected transfer shock loss of young plants at the phase of transferring from agar to sphagnum. And so this is a consideration to keep in mind for further propagation. So overall, we have approximately 100 plants established from 8 maternal lines at the Australian National Botanic Gardens. The spore from the original collections are secured both at the National Seed Bank and at the ATH for further research. And the living collection will be displayed in the new conservatory at ANBG. So excitingly, the propagated plants are now at the stage where they're producing spore themselves. And so this provides a very valuable resource in terms of further research in spore longevity and as an insurance collection going forward. We've developed protocols for short-term storage, germination, propagation and cultivation of this species. And these techniques are suitable for trialling on other significant fern species, at least brown spore species, including those from Australia's island parks. So what next at the seed bank? The trials will focus on understanding spore longevity and trials to understand desiccation tolerance and chilling sensitivity. And doing so, we'll be able to identify the requirements, both in terms of facilities and conditions for effective long-term spore conservation. And on Christmas Island National Park, we'll be focused on increasing and maintaining maternal lion stock from the wild populations to ensure long-term maintenance of the wild population. I'd like to acknowledge the many, many individuals who contributed to this project, and at all stages, a big thank you to everyone involved. And just leave you with further reading of our project and trial conditions, both in the APC Bulletin and Chapter 13 of the Germplasm Guidelines. Thank you.
Thank you very much. Thanks. It's great to be here today at this conference. I would like to add my acknowledgement of the Ngunnawal people on whose traditional lands, from whose traditional lands I'm joining you. So my talk is about seed identification. Identification is particularly important in the biosecurity and weed management areas. And so we have got both the situation of seeds contaminating, for example, seed shipments, but also, machinery that's being imported or being moved around or, coming out of cut flowers. But we also potentially have deliberately mislabeled seed lots if somebody wants to import or move around seeds that they really shouldn't because they're a biosecurity risk or for other reasons. Now, the problem with identification is that there's just not that many ideal options for the non-experts. So there's a limited number of taxonomic experts who can be And there's also a lack of user-friendly and mobile identification tools in general. But for seeds, there's the additional problem. Identification keys for plants are generally optimized for entire plants. So they expect you to have everything available, all the characters, which is a problem already if you're dealing with something that's only in flower and you're being asked for the fruits. But it's much worse if all you have are the fruits or the seeds. And so we end up with situations like this little photo here on the right is part of a set that was sent around a few years ago that was taken off some tourists going to one of the subantarctic islands. And it took people weeks and several institutions until somebody figured out what that might be. So how did I come into the seed identification area? I am a daisy expert in an Asteraceae family, and in 2016 and 17, we developed an identification key, a more traditional one, but a lucid key. Many people will be familiar with this, which is at least electronic and interactive and well illustrated for the Department of Operational Science Support. So this key is actually publicly available. It covers approximately 100 species from a priority list, so they are of weed or invasion concern. And everybody can use that, be they the people for whom that originally developed, or be it other people in Australia or even overseas. And so for those who are not familiar with lucid keys, or again, I assume many people will be, this is what this then looks like. So in the upper left corner, in the upper left windows, you have got a lot of potential characters that are all well explained and illustrated, where you can scroll through whichever character you immediately see and can judge, and you click on it and select a character state. So in this case, for example, in the lower left corner, we see that we've already selected that the fruits are winged, and that we already have selected that they don't have a pappus, so there's flight hairs or spines that you need. And the more characters you choose, the more species disappear from the upper right window, which are the species that are still remaining, that still match the selections that you have made. And so in this case, 6 entities are remaining and 96 are already discarded. And so at some point you can say, okay, I'm now down to a limited number where I can compare the images and I can look at other resources such as species profiles and links for further information on the web. So this has been a very good project at the time, and it was has received very good feedback from the end users. We're using it both for identification and for demonstration and training purposes. But the more I worked on this, the more I realized that there are some limitations with these traditional identification key approaches. First of all, they're very laborious to expand, species by species, collecting all the data and all the information. And the more species we put into there, the more characters we need to differentiate them. You see an example of them there on the right, out of the 36 characters that are already used for those 100 and something species. So the more you put in there, let's assume you want to have 500 species in there, the more overwhelming it becomes to the end user. And the other problem is that even if you try to make it as accessible as possible with good illustrations and explanations, there's no way around the end user needing to understand quite a bit of terminology. I mentioned the pappus earlier, for example, to make sense of the questions that they're being asked. So after I had delivered that project, we considered whether machine learning, image classification with machine learning is now far enough that we can actually use that as an identification tool. So the same way a human expert would look over your shoulder and kind of have an opinion on what they see in front of them, if the computer could at least allow you to reduce the number of options and narrow it down a bit. So the way this works, it's a very hot topic, so a lot of people have heard of that too, but just as a quick overview, you put an image into the model, and every pixel on the image gets put into something like a virtual neuron, you could say, and then you've got a so-called convoluted network where several layers of virtual neurons either fire or don't fire, or fire with a certain strength, to kind of think through what this could be. And at the other end, you get out all the potential classes, it is called in the jargon of the machine learning experts, in our case, plant species, that it could be with a certain likelihood. So you may end up with one species coming out with 99 point whatever % likelihood, on the others with nothing, or it could be a bit more doubtful. So you train the model by showing it lots of example images where you tell it that's what this species looks like, and you may potentially need hundreds of images per class depending on the complexity of the problem. And then you try it out in the field. So we did not just want to try out the AI, we also wanted to develop a system that makes it useful to apply that AI in the field or at the border in situations where people actually need this identification. So we got together with Microsoft and a company, Altus Consulting. So Microsoft co-funded this prototype, this feasibility study, SRO, and Altus Consulting wrote the app interface, and we now have a prototype app with a little model in there for 17 species of wheat seeds that we can actually then show people and say, well, look, would this work for you? And we can try it out in real life scenarios, not just on a supercomputer. So the way this looks like, you can see here on the right, somebody holding that, playing model for me and identifying, in this case, an Osteospermum aglonis seed. So like other apps that are out there, it uses the video feed to give you an identification update with, you can see down there in the case of the Osteospermum aglonis, 99% probability. It tells you how. You can click then on the species name to get species profiles to check against. You can save observation records with, where you are and when you took that photo. And the big advantage here is, of course, if you've got this in this app format and it just looks at an image, you don't need any specialist terminology to get an identification estimate, because it's not somewhere in the cloud. Once this is deployed, you don't have any ongoing cost, and you don't even need network coverage. It's just in that app format as a software on your cell phone. And because everybody has got, well, nearly everybody has got cell phones these days, there's really a great chance of having a wide uptake and applying that in a lot. With this at the moment is, since the applications of this focusing on insect and product farm agriculture. Long-term goal is so that I would like to thank you for your attention.
Hardstaff, and I'm a PhD student at Curtin University, based at the Australian Plant Bank in Sydney. I have been developing cryo-storage protocols for exceptional Australian rainforest plants, including some of the Myrtaceae. The Myrtaceae represent around 10% of Australian native flora and are an extremely diverse family found across the country. Many system engineers and others are valuable for the resources they provide, whether that be timber, food, essential oils, or material for Indigenous cultural practices. However, many Myrtaceae are threatened in the wild by habitat loss, bushfire, and disease. One particularly problematic disease, both in Australia and globally, is myrtle rust. It was first found north of Sydney in 2010, and within 5 years had spread along the entire east coast to reach both the Northern Territory and Tasmania. Because the fungus attacks new plant growth, it can stop plants from producing seed, and repeated infections can kill susceptible species within just a few years. Globally, up to 95% of Myrtaceae are thought to have orthodox seed and can be stored in standard seed banking conditions. This includes eucalypts, bottlebrush, and tea trees. Other species have characteristics that complicate their storage, including desiccation or temperature sensitivities, or limited longevity. These species are sometimes known as exceptional species and require alternate storage solutions. Cryostorage, which is storage in liquid nitrogen or nitrogen vapor, may be a solution for the conservation of exceptional species. Protocols have been developed for a number of species and germplasm types. However, each species typically requires its own protocol. These are 6 of the Myrtaceae species for which I have begun to develop cryostorage protocols. All are found in the rainforests along the east coast of Australia, and all are threatened by myrtle rust to different degrees. Both lemon myrtle and aniseed myrtle have low seed fill, and the seeds of lemon myrtle are short-lived in storage. The critically endangered native guava has freezing-sensitive seeds. All 4 syzygiums have desiccation-sensitive seeds, and the seeds of both lily pillies are metabolically active at maturation, sometimes germinating while still on the tree. All these characteristics make these seeds unsuitable for storage in a seed bank. The first step of developing a cryostorage protocol for exceptional species is to determine whether or not they can be grown in vitro. This includes choosing the appropriate germplasm, determining a sterilization protocol, and optimizing basal media. Cryostorage is a complex procedure with a number of steps, each of which must be optimized for each species. I used a standard vitrification protocol for my experiments, which follows this schematic. Plant material may be treated directly or after encapsulation in an alginate bead, depending on the size and the purpose of pre-treatment loading and plant vitrification solutions is to reduce the amount of free water in the germplasm cells and replace it with cryoprotective agents. This will reduce damage done by ice crystals during both freezing and rewarming. Washing after storage in liquid nitrogen removes those cryoprotective agents, which can be toxic. Lemon myrtle seeds survived 13 of the cryostorage treatments tested, including storage in liquid nitrogen. However, continued growth, such as root or shoot production, was limited. Native guava seed grew into seedlings after all desiccation and cryoprotective agent treatments. However, no seeds survived storage in liquid nitrogen. Aniseed myrtle embryos from 2 treatments that included storage in liquid nitrogen survived. However, continued growth was only seen for embryos that were not stored in liquid nitrogen, which you can see in this figure. I did a number of experiments for Syzygium astral, testing over 30 different treatment and protocol combinations. While some survival was noted for most treatments, contamination was an ongoing problem, and there was no continued growth for embryonic axes stored in liquid nitrogen. I tested both whole embryos and embryonic axes for scaly bark, a species from Norfolk Island, and once again only observed survival and continued growth in material that was not stored in liquid nitrogen. This was another lily pilly for which I did multiple experiments, testing over 20 treatment and protocol combinations. Contamination was an issue once again, and no continued growth was seen for axes stored in liquid nitrogen. Although seed material is preferred for conservation because of its greater genetic diversity, contamination is a common problem for rainforest species. Sterile shoot tips may be a better solution for the conservation of some of these species, and I have begun trials for 4 of my species already. Cryostorage of shoot tips requires the storage of multiple accessions in order to preserve species diversity because of the way that multiplication creates clones of a single accession. Another tool we have found to be of value for the conservation of exceptional species is differential scanning calorimetry. The DSC produces thermographs like these during cooling and rewarming. The peaks indicate the presence of free water in the plant material, with larger peaks indicating more water and potentially more damage. These graphs can tell us whether cryoprotective treatments have been effective and help us to optimize protocols. I would like to acknowledge the funding sources for my project and my supervisors at Curtin University, the Australian Institute of Botanical Science, and Kings Park Science. Thank you for listening.
Good morning, everyone. My name is Rahman Jawad, and I'm a PhD student at Reading University, student as part-time by distance, and I'm working a full-time job as a senior research assistant at the International Center of Agricultural Research in Dry Areas, ICADA, Lebanon. This work is a part of seed quality management project funded by the Crop Trust. My title is Optimization of Seed Harvest Protocols of Gene Bank Accession of the Crop Durum Meat and the Crop Wild Relatives. ICADA is an international organization undertaking research for development, a member of the CIR, and of course, non-profit agricultural research institute. Works particularly in West and Central and South Asia and North and Sub-Saharan Africa. ICADA or Gene Bank was in Aleppo, Syria, but due to the war in Syria, ICADA has been relocated, and now we have two Gene Banks, one in Lebanon for the wild crops and another one in Morocco for the cultivated crops. Collection held in trust by ICADA Lebanon is around 71,375 accession, and the accession are unique, composed mainly of land races, wild relatives, and native species. ICADA has a big number of targeted crops, and we have different facilities in order to maintain the genetic diversity of each accession. The harvesting period at ICADA usually starts at March, end of March. At this period, the clove pack are distributed for each accession. So they are attached to a plastic pack at the beginning of each accession. Harvesting method is manual in order to avoid any loss or mixtures of the material. So it may take a lot of time depending on the crop species. And sometimes you have a variation of the maturation within the trial or within the same accession. So at this period, the daily workers check every day the spikes and collect the mature spikes or pods and collected in the clove pack and left the bag in the field until the whole trial is harvested. Later, then the harvested material goes to the fumigation room for 3 days and threshing and cleaning, and finally to the drying room, 15 degrees Celsius, 15% relative humidity. And this is what it's called the current ICADA practice. The potential problem. Temperature and moisture content are the most important factor that should be controlled to maintain seed viability. And in our case, the harvesting process is taking time to be finished since it's manual and the material are left in the field. Seed longevity can be affected by pre- and post-harvest environments, but controlling factors affecting the seed post-harvest is to a certain extent more feasible. The potential problem is delayed processing of seeds from field or plastic house to the drying room. Seeds are subjected to high and or fluctuation of temperature and relative humidity, which may damage the seeds, affect the quality and longevity of the seeds, so affect the safe storage period in the ICADA Gene Bank. The objective of the research was to examine the effect of delayed processing of accessions of cultivated and wild relatives meat, which show within and between accessions variation in maturity dates by determining how subsequent seeds storage longevity is affected by the progress from the harvest in the field to the drying room. Does the answer vary among the progress, the crops, and should the seeds be removed directly from the field? During the first year, one experiment was conducted in the field composed of one plot of cultivated wheat, Triticum turgidum subspecies durum, and 3 plots of wild wheat, Triticum durum subspecies dicocolis. And the harvested seeds were tested to investigate the hypothesis. 3 harvesting procedures with sequential harvest. The first one, called the immediate, where the harvested material was sent immediately at the same day for 3 days to fumigation room, followed by routine processing. Second one, called the delay processing, where the harvested material placed in separate bag, 1 bag per each harvest date, and kept in the field until the whole trial is harvested, then sent for 3 days fumigation, and then followed by routine processing. The third one is called current ICADA practice that I already explained it before. It's a cumulative harvest and the material left in field until the whole trial is finished. During the harvesting period, we had heavy rain, and it was unusual for our climate in Lebanon. So after the rain, the spikes of the wild plots were shed on the ground. So I collected and called it on the ground treatment and treated as the immediate processing. Since we have this heavy rain, we repeat the experiment using the same procedures and treatments, only we include a new species of wild wheat, Triticum subspecies poeticum. During the two experiments, the measurement in the lab was water activity using the hygrometer, Rotronic, fresh weight, moisture content using the low constant orbit methods at harvest and after drying, the germination test at harvest and after drying, and after drying, the sample were transferred into aluminum foil to the cold room 2 to 4 degrees to be tested using the storage experiment. Storage experiment was done for all the samples. 12 samples were prepared of 100 seeds each. Seeds were equilibrated over a non-saturated lithium chloride solution for 10 days, and the sample transferred into aluminum foil, sealed and stored in an incubator at 45 degrees for different periods and tested for germination. Moisture content was determined using the low constant oven methods at the beginning at admit storage. Here we have examples of the seed survival curves for the different treatments. The red curve are for the immediate treatments and the black one are for delayed processing. And here we have for the current ICARDA practice. Probit analysis applied to the above data in accordance with the seed viability equation by Ellis and Robert, 9080, where V is the viability in normal equivalent deviate after P days in experimental storage, Ki is the fitted initial viability in NED, and sigma is the time it takes for the viability to fall by 1 minute. Here we have the bar chart, the longevity P50 of all treatments in aromatic storage at 45 degrees, 60% relative humidity. This is the result for the first year. This is for the cultivated plot. You can see the green bar for the current, the black one for the immediate, and the red for the immediate, and the black for the delay, and the purple bar chart here for the ground, for the on-the-ground treatments. In the first bar, the cultivated, the estimated P50 for the immediate treatments was either lower or equal to the delayed treatments. The estimated P50 for the current ICARDA treatments was higher in comparison to the immediate treatments for the 4 consecutive harvests and was second best longevity among all harvest treatments. For the wild plots, comparing immediate and current ICARDA treatments, the estimated P50 value seeds for the immediate treatments were consistently lower than the current treatments. Comparing delay and current ICARDA treatments, the estimated P50 for delay treatments from the first harvest, where seeds sitting in the field for the longest length of time, was lower than the seeds from the current ICARDA treatments. Here we have the result for the second year. The estimated P50 for the current ICARDA for the cultivated plot was higher in comparison to the immediate and delay. For plot 2 and 3 that have the Triticum turgidum subspecies Dicoccodus, the estimated P50 for the immediate treatment was either lower or equal to the late treatments. The estimated P50 for the current ICARDA was higher in comparison to the immediate, but lower than with the delayed treatment only in harvest 1, plot 2 and 3. For Triticum monococcum subspecies Vorticum, the estimated P50 for seeds from the immediate treatment was consistently lower than the seeds from the delayed treatments, while P50 for the current ICARDA was the highest among all treatments. As a conclusion, the results obtained for 2 experiments over 2 consecutive years supports the rejection of the null hypothesis because seed storage longevity of cultivated wheat and wild turtles was affected by the different field treatments. Delayed harvest was not a problem. Delayed processing provided better seed quality than immediate processing, and current ICARDA practice does not negatively impact the subsequent longevity of the seeds. On another note, the immediate process for the harvested material was not really immediate since it lasts for 3 days in the fumigation room, then sent to the threshing, cleaning, and drying room. Seed moisture content and ERH were not determined immediately after harvest, so it's difficult to know whether or how the moisture content of the seed changed during fumigation. For that reason, we did an experiment with the same condition as the experiment explained above by removing the seeds and divided into 2 subsamples, one sent directly to the fumigation and another one sent it to the threshing, cleaning, and drying room. And now we are working on this experiment. Thank you for listening, and if you have any question, please don't hesitate to ask.
Hello, hi everyone. I'm Siti Masliza. Today I would like to present my topic entitled assessment of seed quality of indica rice. Rice is a staple food for Malaysia and defining features to Malaysian culture. The quality of seed is vital, as it's able to improve, as it is relevant not only to yield production but also for secure food sustainability. Determination of potential for subsequent longevity is important in order to know the ability of seed in order to generate subsequent crops. In indica rice, CVMR220 is one of Malaysia rice cultivar that are very common used by farmers. Another one of variety is we call it as MR297, or we call it as MR Siraj 297, which this variety much more current compared to the MR220, and this variety is quite resistant to blast disease as well as PMP disease. This variety can be harvested after 110 or 115 days after sowing. The objective of this study was to determine the seed physiological quality, particularly for potential seed storage longevity of contrasting indica rices of CVMR220 and MR297. Seed variety origin respectively from Seberang Perak, which is northwestern in Peninsular Malaysia, and also from Kedah, is north of Peninsular Malaysia. Seed able to germinate normally, speed of germination energy, seedling length, as well as seedling dry weight, and also vigor index have been recorded as a seed quality parameter, along with potential seed longevity. Germination was assessed on a sample of dried seed. Sample were maintained at 15 plus minus 0.5% moisture content. The estimate of percentage germination after different storage period provided observation for survival curve will fitted by probit analysis according to Ellis and Robert 1980. From this present study, it showed the seed survival curve between seed of indica rice CVMR297 and CVMR220, which were produced in Seberang Perak. Maximum value of longevity was greatest for MR297 as compared to MR220. This indicated that the deterioration in subsequent seed longevity occurred sooner in MR220, with P50 only obtained 69 days, followed by MR297 with 116 days. Therefore, this present result indicate that CVMR297 having a longer lifespan as compared to CVMR220. However, if you look into this parameter, it showed that there is no significant difference between the parameter for the MR297 and MR220. If you look into the seed vigor index, even though there is no significant difference occurred, however, we notice here that the vigor index of the MR297 is much more higher compared to the MR220. This result showed the seed survival curve as well as P50 or potential seed longevity data from variety of MR297 obtained from Kedah and also Seberang Perak. It shows that MR297 from Seberang Perak have much more lower in seed deterioration as compared to those from Kedah. Potential longevity of MR297 from Seberang Perak obtained about 116 days of potential longevity value, which is more than double from the one obtained from Kada. Seed lost its vigor as their age, leading to poor seedling emergence, stand establishment, as well as a low crop yield. According to Abdul Rahman and Alice 2009, their report showed that the quality of seed lot of indica cultivar using Aron 1 were comparatively less. In their study, they just obtained only 14.5D and 16.1D. As conclusion, this study suggested that MR297 have greater tolerance to deterioration, and thus its priority strategy for rice seed quality in order to maintain future yield as well as food security. As a recommendation for the next study, further investigation might using more contrasting genotype of indica rices and may use different deterioration tests which can relate with storage seed moisture content. As rice is one of the major crops in Malaysia, such studies will help to improve quality rice seed as it is still insufficient. As reported by Izham, annually Malaysia required about 60,000 tonnes of rice seed only. 53,000 tonnes are produced, leaving a deficit about 7 tonnes. This deficit is currently met by import. That's all from me. So thank you for listening.
Hi, everybody. My name is Nate Kingsley, and today I'm going to be briefly talking about imaging and digitizing the seeds of Hawaii. I currently work in a part of the Hawaiian Rare Plant Program, whose efforts are to protect and conserve the rare plants across the Hawaiian archipelago. There are two main units in this Rare Plant Program, and it's the Micropropagation Lab and the Seed Conservation Lab. I specifically am working in the Seed Conservation Lab, where we have about over 26 million seeds banked, representing around 560 taxa, with most of these being endangered and threatened plants. And we are the largest repository for endangered plants in the Hawaiian archipelago. And about 40% or so, or greater than 40% of U.S. federally listed plants are actually from Hawaii. So we have a high level of rare plants in the lab. So briefly, I was going to talk about one of the projects we have going on in the lab. And what we're doing is we're imaging the seeds that we have so that we can put them onto an online database. Like I said, we've got about 560 taxa or so to image. We're about halfway through. We started in September of 2020, and we're trying to get them all into one large database so that researchers can access these. If there are limitations due to like travel or money, they can look at least at morphological characteristics on this like large database. Right now, we're still hashing out where exactly it should go, but these are some of the ideas I've had. And so what we're doing is, so on the left, you see this before picture, that's just a normal image of the seed. But what we're trying to do is with our Olympus microscope, it's a dissection scope, we're taking images at several focal planes, and we're using software like Photoshop, Lightroom, and others to combine those all into one stacked image so it can increase the depth of field so you can encompass the entire seed in the image. And then we'll clean it up in Lightroom. You can see how much more significant detail you can see on the right side than the left side. We're also trying to take it with different, we're doing a profile picture with one seed, a dorsal ventral, you can't tell as much with this one, and then the variation to see any type of more morphological variation, plasticity amongst the seeds. And what we would also like to do is have some of that morphological data in there as well, so that the research can access it, seed mass, length, and variations of that, of course, because not all the seeds are going to be the same. Awesome. Thank you so much for your time.
Great talks there. We don't have all of the presenters available here to take questions, but we do have some of them. Now I have to get myself organised here. There's questions that have come in on the live Q&A and on the discussion forum, which is great. Let's see what's a good first question. A question for Carolyn Chong from Gemma Hoyle. Following on from your success, you might as well ask that yourself. No, hang on. No. Sorry, I'm confused about what. Lost it. Following on from your success propagating this fern, do you have any plans to investigate maternal effects, e.g. temperature, drought on germination, or longevity of spores?
I think a useful line of investigation might be to ask whether those maternal differences are environmental or have an environmental or genetic basis. If we think about it, this species doesn't occur or occupy a large or obvious environmental gradient as such, although microclimates can certainly differ, so in terms of water availability and other things. So there'd probably be multiple lines of potential investigations. I mean, ideally we would track the survival and performance as such of individuals from earliest stage growth right through to spore bearing maturity and then spore viability. And I think this also, maybe showing my bias, I think it would be also really important to both monitor and maintain the genotypic diversity of both the wild and ex situ collections and populations.
Thanks for that. There's a question here for Lyndall from Emma Sugden. Moving forwards, how might you overcome the contamination issues faced in your trials?
Thanks, Emma. We did try a number of different sterilisation techniques, different chemicals, different time periods, different concentrations. We suspect a lot of the contamination is from endophytes within the plants, so within the seed material. Sorry, that's my cat. So there is potential that we can't overcome some of those issues. They may be important for seed germination in the soil, and then once they're in tissue culture, they really bloom out of control and become an issue. So that's why we think moving to tissue culture for some of those specimens using shoot tips might be a better option.
Another one for you, Lyndall. From someone, this is from Gemma Hoyle, from someone who has no experience of cryo, is it relatively easy to scale up these complex depths for large conservation collections, or is collection size another limitation?
I guess it really depends on the species that you're working with, and in certainly agricultural systems, they are doing like large scale conservation of their germplasm using cryo storage. With rainforest species, it's a little bit tricky because, as Karen and others have mentioned today, getting significant amounts of seed at a time when you're able to process them can be difficult, and some of the seeds only last a short time, so processing them within that short period of time can be quite difficult. But if you can get lots of people involved, that's always helpful. Lots of team members working on extracting material and storing that is definitely possible.
Thank you. There's a question here somewhere that I think Lydia Julia asked you, Alexander, and it's very similar to a question I had myself. My question was more modest in terms of what I'm hoping for. I was wondering how the technology that you've described might be applied to, one of the big genera eucalypts or acacias or something which have a lot of seeds that are, superficially at least similar. And I think Lydia's wants to go one better, and she wants to know, when will we be able to see that you put any seed from Australia, in front of a phone, and we know magically what it is. What are the constraints, and how close are we to that dream?
Thank you. Well, I think there are two different questions there, really. So as far as Lydia's question is concerned, I would obviously love to have that at some point. But it just seems that if you ask where the urgency is, where people really need it and are willing to, invest, I think probably in the first instance, it's going to be biosecurity, because that's a much larger impact than people wanting to identify native plants. And the other question is more about, well, what can the technology do for us? I mean, your own collection, David, as you know, had a student project where you looked at one of those big genera, eucalyptus, where the approach was more, you were spreading out several seeds, whereas the approach that we are taking with this is you may have just a single contaminant in front of you, and you want to look at that. And that, of course, gives you a bit less information, and you've got a whole collection. I guess the thing is, you always have to manage expectations there. Even the best, and in some cases, not even molecular barcodes will always get you a species answer. So I would always try to sell the idea. This is like having your identifications expert look over your shoulder and say something like, oh, I know that's not what that person wrote on the sheet there, or, I can tell you that this is a dandelion, but I don't know which of the so and so many hundreds of species, or something like that. So it already narrows things down for somebody who's not an expert massively, and then they can at least exclude this, which would be really dangerous, or which would be allowed, or something like that, and then they can forward that photo to a relevant expert and help, but only one. I hope that answers.
Yeah, thanks, Alexander. There's another question for Caroline from Bradley Desmond, who I neglected to acknowledge, actually, that Mark, Louis, and Brad are working away as reporters on this session as well. So thanks to those guys for doing that in the background. The question is, given biosecurity concerns, is it possible to reintroduce the ferns back to Christmas Island, or will they be held only as a living collection in Canberra? And then further to that, what other research is planned for the species?
Excellent question. So the primary use of the existing propagated ferns is for them to stay in Canberra. And so originally at the start of the project, we worked through the biosecurity conditions and agreements with DOR. So the use of that and the facility of that is with enough spore stock now, that we now have enough to actually examine and test and understand better the specific conditions for spore storage and to identify desiccation thresholds. And that can feed into, along with the protocols that we've developed, to trials at Christmas Island National Park Nursery to build up capacity and capability there to both maintain and increase collections of spore and then propagate as such in situ on the island to help augment the wild population. But I think also there's a bigger conversation behind that and an ongoing conversation about the ways forward to ensure conservation collections and precedents for threatened species collections, including from Australia's island territories.
Thanks for that. There was a question that came for Rama that was answered on the discussion forum. That's good. There's another one that's emerged for Rama, which perhaps could be taken. I don't think we don't have Rama. So I think that's all of the questions. that's been a really great session, and plenty of good questions there. I've got a few, so yeah, thanks again. I can hear the global applause ringing out, guys. That's really good. The only thing I've got left, I think now, is to thank all of the speakers across the sessions, and I do have a slide. Let me just check to see here. Yeah, no, I'll show that a little bit later. So we're due to finish now at in a few minutes. So we may as well just finish a few minutes early, I think, and we'll come back at 12. Is that correct? Yeah. Great. All right. Thanks, guys. See you in a little bit.
Thank you.